Hi friends, today we are going to discuss about the structure of carbohydrates. Carbohydrates, as all of you might have already studied, that they are of different types such as monosaccharides, disaccharide, tetrasaccharide, pentasaccharide, polysaccharide based on the number of basic units. So first we will go to monosaccharides. So in monosaccharides, they are usually the fundamental units of carbohydrates. So they are broadly classified into two groups, aldoses and ketoses. Aldoses contain an aldehyde group, CHO group, and ketos contain keto group, that is C double bond O group. So here are some examples of monosaccharides familiar to you: glucose, fructose, galactose, ribose. So here you can see that sugars, monosaccharides, are classified to another set of based on the number of carbon atoms present in them. So the simplest is the 3 carbon forms. They are called as trioses. Here you can see example is glyceraldehyde. Next is tetroses, which have 4 carbon atoms. That is erythrose. This is an aldotetrose, and the ketotetrose is erythrulose. And for pentoses have 5 carbons. Example is ribose, and ketopentose is ribulose. Arabinose, xylose, lyxose, all those are pentoses. Now when you come to hexoses, the easiest example is glucose. That is they have 6 carbon atoms and it is an aldohexose. Fructose is a ketohexose. And other hexoses such as mannose, iodose, galactose, allose. There are several examples. Now pentoses can cyclize to form furanoses, and hexoses can cyclize to form pyranoses. Another one is dextro or levo. In nature, the sugars are dextrorotatory, that is they rotate right to when it is exposed to plain light. So that's it. Then comes disaccharides. So when two monosaccharides are linked by a glycosidic linkage, it is called disaccharide. Common examples are sucrose, lactose, maltose. So here you can see the glucose moiety is connected with the fructose moiety by means of this linkage, which is called as a glycosidic linkage. So how it happened is, here was an H and here was an OH. So a water molecule gets condensed and oxygen remains here. Now this is how sucrose is glucose and fructose, lactose is galactose and glucose, maltose is glucose and glucose. Here you can see a peculiar type of bonding. This is called beta glycosidic linkage. This is alpha, this is beta. That is, here both are in the same plane, and here it is one is in rotate. It undergoes a kind of rotation. So this is represented in this way: alpha glycosidic linkage and beta glycosidic linkage. This you will understand more when you study the difference between starch and cellulose. Now coming to more scientific name. Sucrose is called as alpha D glucopyranosyl 1,2-beta fructofuranose. Lactose is called beta D galactopyranosyl alpha D glucopyranose. Then maltose is called alpha D glucopyranosyl 1,4 alpha D glucopyranose. So now comes trisaccharides. When three monosaccharide units are united, it is called a trisaccharide. The perfect examples are raffinose and malacitose. Raffinose is made up of galactose, glucose and fructose. Galactose is connected to first carbon to sixth carbon of glucose. So this glucose is connected to first carbon of glucose to second carbon of fructose. So that is a beta bonding. Okay. So this is it. Then in malacitose it is glucose, fructose and glucose. So that three units are united by first carbon to third carbon, here second carbon to first carbon. Now in this diagram you can differentiate between sucrose, raffinose and stachyose. This is a disaccharide, this is a trisaccharide and this is a tetrasaccharide, stachyose. So here you can see this is sucrose unit. One galactose added forms raffinose, another galactose added by beta glycoside acidics, here it forms stachyose. Now when you come to pentasaccharide, example is verbascose, that here you can see this is a basic sucrose unit, three galactoses added. So all those additional ones are added are galactose. So here you have sucrose, raffinose, stachyose, that is, and pentasaccharide verbascose. So when you come to next category, that is polysaccharides, they have infinite numbers of monosaccharide units connected by glycosidic linkages. The common natural examples are starch, amylose, cellulose, etc. So here you can see this is starch. These are glucose molecules connected by alpha 1,4 glycosidic linkage. So what is the difference between starch and cellulose is this bonding. Here you can see that here it is beta. This is a beta 1,4 glycosidic linkage. Now this is amylose. In this also same alpha 1,4 glycosidic linkage is present. This is present only in animal tissues. So thank you very much for watching the class, and always read further, and in any case doubt you can comment in the comment box. Thank you once again.